And so what we see here is we just have initial activity of beta-galactosidase. What we then did is took a series of our coral-associated isolates, and first we would streak them down the middle of an agar plate, very simple assay, and grew them up for 24 hours. After they grew up, then we simply spotted either our pathogen or our positive control E. coli next to the coral-associated bacteria, and then asked for the ability to utilize the substrate in the media. And here we see that our positive control has lost its ability to produce the beta-galactosidase, and our wild-type pathogen here is producing it, but it's on the opposite from where the coral-associated microbe is. And so potentially now there's something being produced by the coral-associated bacteria that is inhibiting the utilization of this substrate by the pathogen. You can see it's not an antimicrobial because we are having really nice growth of the pathogen. So it's not killing the bug, but it's interfering with its metabolic capabilities. And we went on to show this a little bit more. We still don't know exactly what is being produced by these coral-associated bugs, but they are, we've shown that they can reduce the enzymatic ability of this wild-type pathogen. And through a transposon mutagenesis screen, we've identified a few genes that seem to have involvement in this reduction of beta-galactosidase activity as well. So just to continue the story on how some of these coral-associated bacteria are inhibiting some of these potentially virulent behaviors or behaviors that are essential to get established in the coral mucus environment. One other aspect we looked at was motility. So it's not only that you have to land in the coral mucus, but you have to migrate to an area where nutrients are available and somewhere that you can utilize those nutrients. And so Serratio exhibits two types of motility. The first being swimming. So this is just a stab of the pathogen into a soft agar matrix, and it uses a flagella to swim through. So you can see this spreading of this halo through the soft agar matrix. It also has cilia on the outside of the cell. And so if you spot it onto an agar plate, it'll start to swarm across in this elaborate dendritic pattern here. That's actually quite pretty. Then again, my great undergrad Nick took that same transposon mutagenesis library and looked for mutants that were now deficient in swimming or swarming motility. And so here we're spotting this one. This is one mutant that is deficient in both types of motility here. And so when we spot it in the middle of the swimming plate, it stays stationary. And then also if it's spotted on the surface of the agar on the swarming plate, it doesn't swarm out. Then we added these to our anemone models to see now if reduction in these motility behaviors would lead to a reduction in overall disease prevalence and ultimate mortality. And indeed we see just that. So in here in our wild type, we still see at our highest concentrations 100% mortality. But now in our mutant strain, even at our highest concentration, we're only getting to about 30 to 35% mortality at the same day 7 period. So now we've got reduced virulence of our mutant, indicating that or demonstrating that swimming, at least some sort of swimming or swarming motility, may be involved in virulence behavior. And so we've got indication now that these coral-associated bacteria can alter the metabolism of the pathogen, can alter some of the surface motility. And so we wanted to see if this was true for other organisms, or that swimming and surface swarming were important for establishment and ultimate virulence. So we wanted to go back to our library of coral-associated bacteria to see if they had the ability to not only affect the metabolism, but also affect swarming motility. So here's our negative control. So this is our wild-type pathogen just spotted next to a glass fiber filter disc with no coral commensals on it. And you can see the pathogen is swarming all around the plate in this nice dendritic pattern. Then we tested 6 different isolates of our coral-associated bacteria, again spotting them onto the filter disc, letting them grow up, and then spotting the pathogen next to it. And for 5 out of the 6, we see this clear zone of inhibition of swarming around where the coral commensal is. And I say coral commensal just because we don't know necessarily if it's actually benefiting. That's my kind of default in the realm of symbiosis. Coral-associated bacteria. We've got one example here. This is one on that streak that I showed you of the strain going down the middle. So this one has a clear inhibition on the metabolism, but here we see it's not affecting swarming behavior. So some of these are specialized, and some of these have the ability to affect multiple strategies in our pathogen. So then the final kind of accumulation of this was then to take a community of these quote-unquote beneficial bacteria that either seemed to have an effect on the metabolism or the swarming or swimming behavior, and then what we wanted to do is actually test this in our anemone model to see if we would get reduction of infection or virulence. And so another undergraduate, Ali Alagili, did just that. We took a community of these beneficial bacteria and we tested them either with the pathogen alone. And so again, over a 7-day period, we see our normal progression. As the bacteria is infecting, we get retraction of the tentacles and ultimate tissue necrosis. So this anemone is almost nearly gone. And then in the second treatment here, we first inoculated our commensal strain. So this is a cocktail of 6 of our beneficial bacteria, allowed them to establish on the surface of the anemones for overnight. And this is we did this at a sublethal concentration. I think it was about 10 to the 6 cells per mil. Allowed them to establish, and then subsequently infected our pathogen at our LD50, and then asked if we would get the same level of infection and mortality. And early on, you start to see that the polyps are starting to succumb to infection, but they actually show recovery by 7 days. And so either the pathogen is not able to establish to the same extent, or the bacteria are actually combating it from the start. And by day 7, they look no different from our controls. So this is where grad school stopped, and then I went to California, and we haven't really followed up on this yet. And so this is one avenue that I really want to pick up on now that I'm at Eckerd and have my own space to test some of these things, is then actually go back and see what these microbes might be actually doing. And then especially if we can ever get Aiptasia in a more azenic form, remove the native bacteria, because now we're adding a cocktail of bacteria to an already existent microbial population. And so if we can remove the native population and then add back our selected beneficial bacteria and get a more targeted approach to what these microbes are actually doing. And also, nobody's really studied the microbial community of Aiptasia. So there's a lot of people doing it in coral, but Aiptasia is not as aesthetically beautiful. You can't go to really great places to study them. So not people are studying natural populations of Aiptasia. And so that's one avenue that we would like to explore too. Again, just as a model systems approach, understanding what the microbial community looks like and how it shapes the overall holobiont. So from here, we've got the idea that glycosidases are induced in this pathogen and other pathogens in response to growth and landing on coral mucus. Some of these coral-associated bacteria can inhibit some of these co-regulated behaviors, so the metabolism and surface motility. And together, these bacteria and the opportunistic pathogens dramatically have this potential to alter the host physiology. So either in a good way, or if given the chance, some of these pathogens can really do destructive damage. And so this is kind of one avenue that I want to use to kind of jump off into my new lab at Eckerd and get undergrads studying, again, looking at these interactions, both in vitro, but then in vivo in the system as well. All right, so the last quick little study that I want to give you is a look at the host side of things. So I've been talking a lot about microbe-microbe interactions and just using the host as kind of a proxy to do these, or a model to do these experiments. But what are the hosts doing in response to some of these stresses? And so a lot of the focus in the Pringle lab out at Stanford has again been moving to get Aiptasia recognized as a new model system for coral cell biology and molecular biology, but also understanding some of the biology in and of itself. And so, again, trying to utilize the ability to manipulate and control some of these aspects in the sea anemone system to understand what might be going on out on the field in coral reefs. And so I embarked on a project to look at differential host gene expression using RNA-seq during a coral or a sea anemone bleaching event to try to understand at the host level how the animal is responding to these stressors. So first I want to do a little bit better job introducing the organisms in play. So we have two clonal lines of our sea anemones. One is CC7, derived from Florida, actually, and it comes with an indigenous clade A Symbiodinium. So I didn't mention anything about Symbiodinium taxonomy because it's a nightmare. There's a lot of argument and controversy over it. There are basically 9 clades of Symbiodinium now. 4 or 5 of them have been known to be symbiotic with corals. And so I'm going to leave it at the cladal level here. I don't want to get into species designation, which is more than we need to go here. But basically what you need to know is we've got a clonal line originally derived from one polyp from Florida. We have now propagated that guy over the last 10 years. It is male, so I'm going to call him a guy. We can get it to spawn in the laboratory, so this guy produces sperm. We have another line derived from Hawaii, H2. She's a female, comes with a clade B Symbiodinium that will play into this study as well. So from this symbiotic state, we can artificially make them symbiont-free. So either exposing them to heat, putting them in the fridge for a couple hours, it stresses them out really well, they spit out all of their Symbiodinium, or just propagating them in the dark and inhibiting photosynthesis. Eventually they will kick out all of their symbionts. Until we have confirmed aposymbiotic animals, then what we can do is we can take some of our cultured algae. In the case of SSB01, this is that clade B that originally came from the Hawaiian strain. So we can isolate that strain, grow it in the lab, and then reinfect it into aposymbiotic CC7 animals. And so now this is the same host background as our original strain, but now contains a novel clade B Symbiodinium that it does not normally associate with in the environment. And then we can ask questions about thermal tolerance and overall physiological differences between now having either its endogenous clade A strain or now this new novel clade B. And of course, within the realm of symbiosis and model systems, this is what we're trying to mimic, right? So we've got our healthy, very vibrant reef, lots of colors, can undergo bleaching in response to thermal stress and other stressors, and then we can basically mimic that in our anemone system by exposing these anemones to similar conditions. And then again, relying on the native autofluorescence of the chlorophyll, we can monitor the status of that symbiosis in terms of number of Symbiodinium. And of course, this is a qualitative look, just looking at stereofluorescence microscopy. But we can also do these experiments where we acclimate our anemones for a given time. We keep them on a 12-12 light dark cycle, expose them to our 34-degree heat stress. This is our standard heat stress that elicits bleaching response in these guys. You may think initially that that's hotter than what corals are normally experiencing, which is true for some areas. But in our field site in American Samoa, these are corals that are living in very shallow lagoons and at low tide can experience 34+ degrees and still be okay. So this is within the realm of appropriate temperatures and it's enough to warrant bleaching in our sea anemones. As we're doing this heat stress event, we can take these anemones out, homogenize their tissue, and then run that homogenate through a flow cytometer. And then again, using the fluorescence of the Symbiodinium, gate them out and get a really accurate and precise count of how many symbionts are remaining in the anemones, and then normalize that to overall size of the anemone to get a relative sense of how much bleaching is going on. And so if we look at relative bleaching in our 2 strains that we tested here, so in orange here is our CC7 with its endogenous clade A, so this is the normal association that we see in the environment. And then the green line here is that CC7 clonal line with that clade B Symbiodinium, so that novel combination. And in response to the same level of heat stress, we see that our endogenous combination is more thermally tolerant, so it bleaches less over a 4-day period after exposure to 34 degrees, whereas this novel combination is much more thermally sensitive. And so this is reminiscent to what we may see in the field. Some species of coral, even if it's the same species, may harbor different strains of Symbiodinium. And if you look out on a reef, you may have one colony that fully bleaches sitting next to a colony that looks completely healthy. And so people in the coral field are really asking now, what is driving these differences in thermal tolerance? Is it differences in host genotype? Is it differences in the symbiont genotype? Or is there something else coming into it? And so what we wanted to do is take a transcriptomic approach to get at this question and to ask what are the global gene expression differences in Aiptasia under heat stress in our different strains. So we can test either the native strain combination with the host and its endogenous clade A. We can test this novel combination, and then we can also test it in the aposymbiotic state. So that allows us to ask, without a symbiont present at all, what is the expression pattern or what is the response from the host itself in response to this stress, and then what may be coming in from the symbiotic status. And can we find genes or expression patterns that are actually correlated with the onset of bleaching? So is there expression patterns that may be indicative of the bleaching pattern that happens? And then are there differences between the strains? So we see a difference in thermal tolerance at the phenotypic level, measuring the number of symbiodinium that remain. Do we see those similar differences at the transcript level? And so our experimental approach was to take our endo strain. So this is our novel normal combination of CC7 with its clade A. The Sym is going to be our novel combination, so now with our clade B, and then also our aposymbiotic strains. And so we typically grow them on a 12-12 light dark cycle at 27 degrees, and then either kept them at 27 degrees or exposed them to this heat stress, and then sampled throughout that heat stress for both measurements of bleaching, but then also preserved them in RNA later, and then extracted RNA and did RNA-seq. And so we analyzed differential gene expression using RNA-seq and then the package DE-seq2 to actually call differential expression, and then again quantified the number of symbiodinium remaining by the flow cytometry that I showed you before. And now because we are sampling across a temporal timescale, we wanted to look at global gene expressions over time to see if there were clusters of genes that correlated together and showed different expression patterns over time. And so we used a program called STEM. This is short-term expression miner, originally designed for microarray experiments, but again incorporates time series expression data and clusters genes that are behaving the same way in a temporal pattern. And from this we got, how many plots are up here? 10? 10 different clusters of significant expression profiles. And so we see that some go up early in response to stress and come back down. Some go down. Some do up downs. And so I don't have time to go through each of these profiles, and also we don't understand all of the genes that are involved in each of these profiles. But I want to focus on a couple that seem to be showing us some interesting stories between our different strains. And so when I first start going through this, I'm going to focus on the more thermally sensitive combination. So that's the CC7 with the clade B, the one that bleached faster, because again, we get a little bit more dramatic of an effect due to that thermal sensitivity. So this first group that I want to talk about is genes that go up really early. So they're fold change, high fold change expression at 3 hours in response to this heat stress, and then they come back to baseline by about 12 to 24 hours. And so in this cluster, we have about 1,100 genes. What I'm plotting here are the 10 most upregulated at this 3-hour time point, just as a representative. But again, all genes in this profile may not be to this magnitude, but are all showing the same temporal expression pattern over time. So all of these genes have an upregulation at the early time point and then come back down to baseline or are even downregulated as the time goes on. And so initially, those of you that have done any sort of thermal stress on any system are probably thinking that these are going to be the chaperones, the heat shock proteins, and you're absolutely right. So we've got heat shock proteins, we've got caspase, so we know that apoptosis has been implicated in coral bleaching as a mechanism for cell death and thus releasing the symbiodinium. We also have a lot of TNF associated factors and TNFR, which again have also been implicated in other coral studies looking at heat stress. So, but what was interesting to us is that, so not a lot of coral studies have focused on these really early time points because oftentimes they're natural bleaching events and they go out, once you observe bleaching, now you're either into the bleaching event, and that's when they start to see these proteins. But what we see is that they're really up early in response to the stress, but then come down before we actually see bleaching. So if we look, we don't really start to see bleaching until significant bleaching between 24 to 48 hours in our heat stress. And by that time, these genes are way back down. And so the initial question was, well, are they correlated with bleaching? is there a long-term kind of effect of this? So do we get this spike and then the message hangs around and ultimately leads to bleaching? Or is this a very just generalized stress response that happens in the organism and happens in a lot of organisms in response to stress, but are not necessarily tied to the bleaching phenomenon? So again, because we've got the benefit of using the Apo condition in this scenario, we can test how the gene expression in Apos look compared to the Sym, again, without that presence of the dinoflagellate. And we see the exact same pattern. And so in the host alone, without the presence of symbiodinium, so organisms that can't bleach, we still see this upregulation at this early time point within this set of genes. And so this led us to think that maybe these set of genes are not really related or correlated with the actual bleaching that we see. So then we wanted to look at another group that are up early but then stay up. And so these initially seem to be more reminiscent of genes that we might anticipate being involved or predictive of the amount of bleaching that we see. And indeed, when we look at the Apo scenario, we don't see the same sort of dramatic effect. So we see some maybe upregulation, but this is now only a log 2-fold of 2, whereas we're getting log 2-fold of 4 to 6 in some of these genes that then stay up. And some of these genes are kind of at almost baseline for the entire. So now we've got something that's different between our two symbiotic states. So then we went into our database and tried to figure out what these genes were. And the top 60% of the top 50 genes were uncharacterized or had no annotation. And so, of course, that's a little bit frustrating, but also kind of exciting in that this opens up the door to a big window of genes that may be actually doing something, but we have no idea what, just because they're unannotated. And so we've really got more work to do now to go in and figure out what these genes might be. Another significant proportion of these top genes were transposable elements and retroviral proteins. These again were observed in a study of Orbicella species back in 2008 during a thermal stress event. And so again, this is something that is what people are finding, but again, we really don't know exactly what these genes are doing and if they are involved in bleaching, and if so, how might they be involved. So here's just kind of a top list of what these are. So a lot of retroviral genes. I didn't include all the ones that have no annotation because that would just be a long laundry list of NA. But again, really not much idea in terms of the actual biology in relation to bleaching, what might be going on in this pattern here. So right now I've talked about the thermally sensitive strain. So how does that thermally sensitive strain and its gene expression compare to the more thermally tolerant and endogenous combination? And so I'm going to show you a few heat maps. Here's that cluster of genes that is up early. So this is 0 hours, 3 hours, 12, 24, 48, 96. And so here's that cluster of genes that are up both in the Apo state and our novel combinations. So at that 3-hour time point, you get this massive upregulation, and then this same cluster of genes is then back down to baseline as we continue. If then we compare our thermally sensitive strain to our thermally tolerant strain in the same cluster of genes, we see a slight upregulation in these genes, but it's